Hello everyone. Good morning, good afternoon, good evening to all. Dear authors and invited guests, welcome to technical session 8C. I am Riddhi Gohil, moderator of this session. On behalf of Global Knowledge Research Foundation, it is my great pleasure to welcome you to the 6th World Conference on Smart Trend in System Security and Sustainability through Zoom. I hope you will enjoy the knowledgeable interactive session through the day. So there is a special note for the all presenter. You will be given a 12 minutes for the question answer and the 3 minutes for the, you will be given 12 minutes for the presentation and 3 minutes for question answer. And on 10 minutes, I will raise a gentle reminder. There is one another request to all. Please stay connected with us until the closing remarks. And if you have any query and updates, please let me know in the chat box. So just before we get started, I would like to introduce to all our session chair, Professor Gitanjali Shinde. Hello, ma'am. I welcome you, ma'am.
Hello. Thank you.
So Dr. Shinde has overall 40 years of experience, presently working as a SPPU approved assistant professor in Department of Computer Engineering, Vishwakarma Institute of Information Technology, Pune. She has presented a research article in a World Wireless Research Forum meeting, Beijing, China. She has published 50 plus papers in national, international conferences and journals. She is the author of 10 plus books with publishers Springer and CRC Taylor and Francis Group, and she is also editor of book. So, with your consent, ma'am, we will start our session.
Yes, we can start.
So, I would like to invite our first presenter, Miracle, and the paper title is The Potential of Deep Learning Techniques for the Classification of SARS-CoV-2 Variants Based on the Genomic Sequence Information. Yes, you can start your presentation, ma'am, sir.
Yeah, I hope everybody can hear me.
Yes, sir.
Okay. Just a moment, please. All right, then. Good day, everybody. My name is Miracle Mesomachinwe, and I'll be presenting on the topic, the potentials of deep learning techniques for the classification of SARS-CoV-2 variants based on genomic sequence information. Now, this is my outline. I'll be giving a brief introduction, and then go over to the aim of the study. Then we'll talk about molecular techniques for identifying SARS-CoV-2 strains and variants, and then the deep learning techniques for classification. It's not common knowledge. The SARS-CoV-2, which is severe acute respiratory syndrome coronavirus 2, was identified first in Wuhan, in China in 2019, and it has been a global health concern. And specifically, the World Bank in 2022 released data for alarming figures, over 200 million cases that led to 5 million deaths. And over there, the right-hand side, it's just the schematic representation of the SARS-CoV-2 structure. Now, genome and genetic mutations, however, have been playing a vital role in the morphosis of coronavirus strains and even the variants. And it's very important to take note of the fact that these mutations lead to different morphs or different forms that are resistant to drugs and vaccines and all. Basically can be classified as strains or variants depending on their genetic makeup and their phenotypic expression. Now, when we talk about strains, we mean that the fact that the organisms that are under consideration are distinct or very evolutionary distant, but belonging to the same family. Now, that's what strains are. And also the phenotypic expression is marked. That is, it is very observable. It is something that you could say, okay, this strain exhibits this kind of phenotype, and therefore it can be classified in a different strata. Now, variants on their own are very closely related to one another, variants of a particular organism, such as the SARS-CoV-2 variants. It is very kind of hard to distinguish when you look at their genomic sequence information. Now, the next slide shows the different strains of SARS-CoV, of coronavirus, and also points out the fact that the similarity, genomic similarity between the SARS-CoV-2 and the MERS-CoV, which is the Middle East Respiratory Syndrome coronavirus, is the genomic sequence similarity is 50%, while that between SARS-CoV-2 and SARS-CoV is 79% similarity with regards to genomic sequence information. Now, what about the Delta and the Delta variants? I've talked about the strains. Now, the variants and the variants of coronavirus actually are more similar when it comes to their genomic sequence content. Now, present study has shown the use of deep learning actually to be able to identify or to distinguish SARS-CoV-2 variants from other similar viruses, and even within the coronavirus family. And it's important to take note of the fact that when we can identify the particular variants of concern that a patient is suffering from, it's easier for clinicians to be able to administer drugs on time, you know, to ameliorate such cases, maybe cases of development of resistant strains when you treat the wrong variants. Now, the aim of this paper was simply to review the success that have been achieved in classification or identifying of distinguishing of SARS-CoV-2 strains based on the genomic sequence information. And specifically, we tried to look at studies that have implemented deep learning techniques. Yeah, and the molecular techniques that are on the ground presently that are used for the identification of SARS-CoV-2 strains and variants, basically, they are the reverse transcription quantitative real-time polymerase chain reaction, which is the RT-qPCR, and also nucleic acid sequencing. We'll talk about the next generation sequencing and all. And these particular techniques focus on two key parts of the genome of the SARS-CoV-2, which are the ORF, Open Reading Frame 1AB, and the N gene. Now, with regards to deep learning techniques for classification of SARS-CoV-2 variants, we present just a few of the data that is in our review. And the first was by Saha et al. In 2021. Saha et al. Actually carried out a research that was quite remarkable. They tried to develop a tool which they termed, or a software which they termed the COVID predictor. And the COVID predictor was based on the memory network, and it was used as a discriminatory tool for distinguishing between SARS-CoV-1, SARS-CoV-2, between SARS-CoV-2, SARS-CoV, dengue virus, viruses basically that have genomic similarity with the SARS-CoV-2, basically was what they tried to investigate. And they actually had a 99.5% rate of accuracy, 99% accuracy, basically, with their model. And also, a very outstanding work also was published by Diff et al. In 2021. Diff et al. Investigated the use of LSCM, which I've spoken about the full meaning, and the gated recurrent units. Now, these two different models are actually forms of recurrent neural network. There are different types of recurrent neural network. And they were used to distinguish the different types of different strains of the coronavirus family. And one striking thing was the fact that this technique actually does not have an alignment phase, you know, like the blast search. Blast search, what it does basically, when you put your sequence there, for it to be able to tell you that it's one of the variants or not, it has to carry out the sequence alignment first before predicting what type of organism you have there. Then the work by Wata and Chimedza in 2021 actually also used they used convolutional neural network in association with bidirectional LSTMs, and they tried to classify SARS-CoV-2 and tried to classify genomic sequence as either SARS-CoV-2 or otherwise. So it's very remarkable also, the research. And the data based on the performance metrics they evaluated was 99.5% accuracy, sugar classification accuracy, and the AUC was 100%, which was remarkable. All right, so these are some of the data that we extracted from literature. Now, in conclusion, the survival of species has been vastly predicated on the ability to mutate, basically. Now, when we are able to detect on time the type of or the exact variants of SARS-CoV-2, clinicians have the knowledge or the knowledge base of heuristics to be able to predict or know the best line of treatment for patients. Yeah, and it's important also because of the fact that most of the studies that have ensued, most of these studies actually have focused on the use of deep learning for classifying SARS-CoV-2 strains, and most ignore the variants because of how the genomic sequence is very, very similar. You can see that even a point mutation can cause a marked phenotypic expression and increase virulence of a particular variant. So this research basically this particular study or review was to spur up interest in carrying out research that has to do with use of deep learning for identification of the SARS-CoV-2 variants themselves. So I'd like to acknowledge the World Conference on Smart Trends in Systems and Security and Sustainability for this opportunity. I'd like to acknowledge Capicase and Covenant University and also Landmark University, for the opportunity to present. Here are my references. Thank you very much.
Hello.
Yes, hello.
It was a nice presentation, and I can see that this is, I think you are at early stage at your research. So you have come up with the background study about why genomic sequences you are going to study further using deep learning. So my few questions are like, in this field of identifying the SARS strain using the deep learning, why you are thinking about genomic sequences? Why you are not thinking about other strategies?
Okay, basically I am thinking about using genomic sequences basically because of the fact that I'm into bioinformatics research and specifically functional genomics research. And what we basically do is we try to see how we could apply knowledge of computer science, computational knowledge to solving biological problems, basically. And genomic sequence information from the trends in literature will I have been able to identify the fact that the long short-term memory, the LSTM models, which are basically used for speech predictions and it basically incorporates text data. You would see the fact that the genomic sequence is in form of text data, where you have the ATCG. And yeah, why not exploit this kind of feature for predicting mutations? So that's pretty much what I'm doing. Yeah, that's
And
why I'm
about,
focusing on genomic sequences. Yeah.
okay, okay. And about deep
Excuse me.
learning models, yes, audience, if you're having any questions.
Hello, sorry. Can I also respond to that question,
Yeah,
moderator?
yes, yes, yes,
Okay.
please.
Thank you. Miracle is actually one of my supervisees, and so I think to respond to your question, I think we actually look at genomic analysis because, you know, these days we try to treat patients based on their genomic makeup. And so we don't want to generalize treatment for patients anymore. So predicting a patient's treatment based on their genomic makeup is the essence of this research. So if you use genomic sequence, you can come about a way of treating every patient based on their genomic makeup. And that is the essence of using genomic sequence analysis for this deep learning, you know, work. Thank you.
Okay, okay. Now I would like to know that as deep learning models, there is the black box, like the how it is trained. Uh, for that, which technique you will use to give what is actually this deep learning is having, like the explainable AI. So how you are going to explain that your deep learning model is based on which part? Because as it is about genomics, so what are your views on this?
Oh, yes, thank you very much for the question. Recently I carried out a research, yet to publish, so I may not be able to speak on the data that I've gotten so far. We're able to exploit the genomic sequences, carry out feature extraction, with respect to the fact that we only measured on the single nucleotide polymorphisms. So with this, we're able to design a gene budding based technique, basically, that actually characterizes or is comprised of the different mutations in different genes. So the order of these mutations in the genes, we use it to predict the resistance status of strains of a particular organism. You understand? So a particular organism based on genomic sequence could be resistant to a drug, or susceptible or sensitive to another drug. So we tried to, in a different study, basically, we tried to look at this. And with regards to this particular study, we're trying to come up with a more sensitive technique that could detect these point mutations that ensue or that come about or come to play in SARS-CoV-2 variants, basically. So that's still in the pipeline.
Okay, okay. Can you please go to slide number 9?
Okay.
Yeah, you have done very good, thorough literature work. Just one doubt is that in third research work, they had
Yeah.
given 99.95% of the classification accuracy. Then what the difference you are going to give from your research?
Yeah, thank you very much. What they actually did was, like I've been saying, they tried to classify the strains of SARS-CoV-2. Research has not the variants. When we talk about variants, I mean the Omicron, like the popular ones, Omicron variants, the Delta variant. You see, most research has not come up to try to identify or to classify the different variants, or to say, okay, this particular organism is the Delta variant, this particular organism is the Omicron variant. So that's what we're trying to do, developing a more sensitive technique that can detect point mutations.
Okay, got it. Very good. Have you gone through, like, is there any authors they are actually working again on the same topic, or they have already done for classifying these Delta variants? So for that also you can go for means you can have that literature survey for your research.
Yes, yes. Basically, I did a search this morning while preparing for this presentation, and I see so that there's no single publication where deep learning was used for classifying variants, SARS-CoV-2 variants. And that's why this topic is very important and crucial to put it up to the academic population or community that this has not been done. Looking at how can the work that has been done basically be upscaled to detecting the variants. Now, that's the potential of these deep learning techniques for classifying SARS-CoV-2 variants.
Okay, so nice presentation and all the best for your research. That's it from my side. Audience, if you are having any questions, you can unmute yourself and please ask.
So nice presentation, sir. So we will forward to our next presenter, Marion, and the digitization techniques for the representation of genomic sequence in LSTM-based model. By the way, instead of Marion, Abnego will be presenting this paper.
I think it's having issues connecting.
Yes,
so
sir. I think so.
I don't know. Can I stand in for him or something?
Sorry,
because we're in the same research cluster and we're co-authors.
Okay, no problem.
Okay. All right, so I'll just go ahead and share. Okay, so to avoid the delays out, I'll just go ahead and present.
Okay.
Okay. So good day everybody once again. Yeah, my name is Miracle Mesomachinwe, and I'll be presenting on the digitization techniques for the representation of genomic sequences in long short-term memory-based models. Now, this is the outline of my presentation. And by way of introduction, in all life forms, the genetic information is stored in the form of either the DNA, which is the deoxyribonucleic acid, or the RNA, which is the ribonucleic acid. The DNA is distinct from the RNA, not only based on the basis, but as computational biologists, the bases are kind of important in this particular study. They are also distinct with regards to their structure. The DNA double helix, RNA single stranded, although RNA has been identified to come in primary, secondary, and tertiary forms. And genomic sequences basically are usually written in the 3' to 5' direction when it is spoken of in the primary order of sequencing, primary order of nucleotides. And the genome of organisms basically is arranged in the order J1, J2, J3 till Jn, where N represents the length of the whole genome. Now, the J represents the genes, gene sequence, introns, and the exons of organisms. Now, J four different characters, which are the A, C, T, and G. And these characters basically represent the nucleotides that are incubating the genomic sequence, basically, or in the genes or introns and exons. Now, advances in high throughput sequencing has led to big data, and big data with respect to genomic sequences that have proliferated in the scientific community and that are in databases. Now, digitization itself is a computer science term that is used to depict the representation of the genomics, like the nucleotides that I just spoke about, the ACTG, in the form of computer readable, in computer readable forms, basically. And the particular deep learning algorithm that we focus on in this particular study is the recurrent neural network. But deep learning basically has been used for a multiplicity of work. It has been used in speech recognition, in text sequence prediction, image classification, in natural language processing. Various deep learning models have been used, like in image classification, convolutional neural network is used. The recurrent neural network. Now, recurrent neural network is the particular or the specific deep learning technique or algorithm that we considered fully in this work. The specific subtype of recurrent neural network, there are two different types, the LSTM, which is the long short-term memory networks, and the gated recurrent units. But we are specific about the LSTM, the long short-term memory network. Now, this study sought to extract and explain the various digitization techniques that have been employed by various authors, or by authors in different literature, and that have been based on the long short-term memory network. Now, just by way of overview of LSTM, LSTM basically has been used for the particular inputs for LSTM, long short-term memory network, is text data. Text data, when I talk about text data, I mean sequence of words. Now, it's very fascinating how computational biologists have been able to apply computer science to biological problems and represent biological terms in machine learnable forms. That's what we're going to be discussing. This is basically the functional structure of the LSTM network, which has 3 gates, which are the forget gates, the input gates, and the output gates. Yeah. All right. So the different digitization techniques that have been reported in literature include the codon indexing, the KMI embeddings, the bag of words, one-hot vector encoding. It's very remarkable to note the fact that the genomic sequence has been represented in form of indexes or numbers. What we can see in this slide shows the genetic code, but it's like an adapted genetic code where U has been replaced with T, because this particular study by Khan et al. Assumed that the sequence was made up of DNA and not, they had DNA sequences. So they had to try to come up with a way to represent the genomic sequence in the form of numbers. So as you can see, the genetic code had unique index attached to it. So instead of lysine or whatever amino acid, so it was represented with numbers. Also, this work by Mohammed et al. They use genomic sequences also, and using the separator, just a common Python separator, they were able to convert the genomic sequence to a sequence of words. All right, so, and they created these indexes. All right, the KMA technique also, I would illustrate that with this image, where the genomic sequence is converted to different, it splits to a format, or just in any KMA, any number, is any specific number of sequences. For this particular study was a format split sequence. Now, also one-hot vector encoding has been used to represent genomic sequences also. So this was reported by Cheng et al. 2022 and Zhang Zi in 2016. Now, in conclusion, we have identified the different LSTM models that have been used for representing genomic sequences in computer readable formats, which are 1, 2, 3, and the rest. All right, so we also identified the fact that most of the techniques in literature use the CNN as a feature extraction mode, where they use one-hot vector encoding. So I would like to acknowledge once again the World Conference on Smart Trends in Systems and System Security and Accessibility, CAPIC ACE, Covenant University, Landmark. Here are my references. Thank you.
Nice presentation once again. And my question is like that. Why we are going for digitization? Because there are many threats. So have you read about or have you done any thorough literature about threats that can be there when you are going for digitization of the sequences?
Sorry, I didn't get your question. I didn't get your question.
Like we are doing the digitization. So
yes.
Are you aware of the different type of threats that can be there?
Of threats?
Yeah.
Threats. Please be specific. I don't get threats. When we talk about genomic sequence is basically a sequence of A, T, C, and G that represents an organism's genetic makeup. Yeah, so and basically it's computational. We're just trying to, how can we represent this genomic sequence that is vast in a more computationally less expensive way. So that's why scientists have come up with these various digitization techniques that we have elucidated here in this study.
Okay, so among these techniques, whichever you have studied, which technique is the efficient one based on the accuracy and based on the space it is taking?
Okay. The technique that is most efficient is relative, because the digitization technique that is used in literature depends on what you're using as your machine learning model or your deep learning model. So we noticed that a lot of literature, the ones that had to do with CNN, LSTM methods, where they use CNN as the feature extraction tool. Those particular literatures employed one-hot vector encoding. Now, literatures that basically just went straight to the point and used LSTMs, they employed the code, the one that has to do with indexing. Yeah, the derivation by indexing, that was what they employed. So it is basically machine learning or deep learning technique oriented. So it depends on the deep learning technique you're using. Decision technique that you use depends on what deep learning technique you're going to be employing. That's basically it. So I don't think it affects the accuracy or the precision or whatever. Yeah.
Okay. And about your research, like which data preparation you need to do before building the LSTM or CNN models?
Okay. Which data preparation? There are lots of data preparation tools. There are lots of data preparation tools.
I mean, are you working on real data or you have taken
yes.
somewhere?
I basically have worked on quite a few, not one, not quite a few. But I would mention one that has been very, very unique. And that particular method employed the single nucleotide polymorphisms, basically. So instead of considering the whole genome, we try to restrict our mind. Like when you're talking, when the organism in focus is an eukaryotic organism. Eukaryotic organisms have a very, very large genomic sequence. They have genomic sequences that could span up to millions, as opposed to viruses that could just be kilobases. And so with those kind of eukaryotic organisms, even human beings, we try to use a method, data pre-processing technique, first of all. That actually is one of the tools we use in our next generation sequencing pipeline. In that pipeline, we carry out a method called variant calling. Now, in variant calling, we extract the different mutations that are in the strains or the different submissions to databases as opposed to a reference. Again, so the reference serves as the template by which we decide, okay, this particular point, there's a mutation. So all the mutations there, we characterize them. And then we use any of these methods, basically. The research I carried out, I used the CNN method, the CNN LSTM method. And so I converted the SNPs. The SNPs are still the ATGC sequence. Yeah, so I converted them to sequence of numbers based on their genes. And then the CNN was able to read the features and make my predictions, basically. Yeah.
A nice presentation and all the best for your research. Any questions from audience?
Thank you so much for the great presentation.
Thank you.
Yeah, we will moving our next presenter, Prashant Vats, and the paper title is OCD, On-Demand Ordering Food Through Online Crowdsourcing. I think he is absent. So we are moving our next presenter. Ariana Tolmack, and the paper title is Hybrid Art as a Result of Synthesis of Art and Technologies.
Greetings to all participants. Let me start my presentation. My name is Marina Tolmach. I am from Kyiv National University of Culture and Arts, from Kyiv, Ukraine. And let me present you our study about hybrid art as a result of synthesis of art and technologies. And let's go. In the modern world, ICTs have become an invariable attribute of human existence, influencing and determining almost all areas of his life and transforming entire industries and society in general. Technologies becoming more and more accessible are transforming the economy and politics, culture and arts, science and leisure. In particular, technological progress changed the face of contemporary art and became the starting point for new types of art, the characteristic features of which is hybridity as an important principle of modern art. Hybridization of art and technology is one of the most science and consequences of more complex processes of cultural hybridization. As a result of cultural hybridization, hybrid art is being formed as a direction in which artists realize their artistic tasks, interacting with related fields of science and new technologies, in particular with biology, robotics, physics, experimental interface technologies, language, gesture, face recognition, artificial intelligence, and information visualization. The works of the hybrid art have a research basis but also address emotions and make it possible to think and feel. Thus, they get aesthetic value. Also, the hybrid art shows a change in thinking not only of artists but also of scientists. It's often difficult to distinguish scientific and technical research from an artistic project because of the synthesis of the science and art. The hybridization of art and technology, the synthesis of the artistic process and scientific research led to the emergence of the science art or arts and science, in which scientific experiments and technological innovations are the dominant principles. An illustrative example of science art is the mind-controlled Cybergrouch project of the 18 Apples art group, which aims to investigate the problems of transferring human consciousness. Electrodes were implanted in the cockroach's tactile organs and chest, after which they tried to control the cockroach with the help of electrical stimulation of neurons and the brain-computer interface. Such a metaphorical transfer of human consciousness was intended to clarify the nature of fear of this possibility, fear of the latest technologies for controlling the human brain in general. Such experiments actualize the ethical issues of power, identity, intelligence, and society, to some extent distinguishing hybrid art from aesthetic art. Its ethical and religious aspects objectify the collision with artistic and cognitive values because it's an aesthetic spectacle. It popularizes and devalues science. Attempts to understand the works and find out the specifics of hybrid art are made using the concept of algorithmic and fractal aesthetics. Thus, thanks to algorithmic aesthetics, the creative process and works of art are considered using computer technologies, methods of applied mathematics and cybernetics. Researchers pay attention to the possibility of developing an aesthetic system for the algorithmic specification of aesthetic points of view. It is believed that aesthetic judgment consists of a set of interpretive conventions and criteria for evaluating art, but it can be different for different people. So there is a need to concretize various thoughts in terms of the homogeneously structured system of algorithms. Algorithmic aesthetic involves using algorithms that generate descriptions, interpretations, and evaluations of artworks to investigate the problem of aesthetics. Fractal aesthetic is directly related to the study of self-organization of complex non-linear dynamic systems, primarily in mathematics and natural science. Fractal geometry is related to the description of infinite self-similar structures arose in the result of successive repetition of geometric or algebraic transformation, the basis of the so-called fractal arts. One of the exceptional features of fractal art is the visualization and sensualization of infinity, not as a mathematical abstraction, but as a mechanism and result of the construction of an artistically mediated reality. Therefore, the works of fractal art are characterized by a specific type of artistic imagery, in which the construction of extremely beautiful, complexly organized, impossible spaces is possible. Therefore, the works of fractal art are we can see in Da Vinci Turbulence and, for example, Hokusai's Great Wave and Pollock's paintings. Also, fractal patterns repetition is common in the art. It presents an artistic challenge. For example, many have tried to fake Pollock's fractals, but they have all failed. The specificity of the creation of fractals by artists prompts us to think about the question: what determines their aesthetics? Automatic unconscious mechanism inherent in the artist's biology, or intellectual and cultural factors. In Pollock's case, experts believed his fractal aesthetic is the result of an intriguing combination of options. Pollock's fractal patterns derive from the movement of his body, yet he spent years consciously refining his field technique to increase the visual complexity of his fractal patterns. One of the types of hybrid art is bioart, or transgenic, biological, or biotechnological genetic art, as a result of artists' interest in biotechnology. Works of transgenic art mostly illustrate or model genetic and evolutionary processes. This wide range of work, ranging from paintings, photographs, and sculptures, depicting segments of DNA, fragments of chromosomes and viruses, various installations that convey the artist's perception of their structures, genetic self-portraits, and ending with project created with the involvement of genetically modified organisms. One of the most famous artists is Eduardo Katz, the author of various artistic practices, and including performance, poetry, and holography. As a bioartist, based on his own words on the application of the method of appropriation, he used biotechnology in his works to influence the genetic structures of animals, plants, and himself. His most famous and controversial work is the chimera animal Alba. Employing molecular biology, Katz combined jellyfish and rabbit DNA to produce a bunny that glows green under blue light. At one time, the appearance of Alba caused controversy in the world, actualizing the question of the artist's responsibility for the work, the limits of creativity, the future of humanity and nature. But today, the image of the green rabbit has become a cultural symbol. Another transgenic creation of Katz is the flower Edunia, a genetic hybrid of the artist and petunia, or plantimal as the artists call it. In the red veins of the flower lives the immunoglobulin gene of Katz. Such a combination, in his opinion, symbolizes the common nature of all living things. The work of works of Katz is not just a demonstration of the achievements of genetic engineering or an opportunity to enjoy the company of an artistic chimera. It is an example of the rapprochement and kinship of man with all living creatures of the planet. If you know, microbiologist Alexander Fleming is considered the founder of transgenic art. Fleming is widely known as the brilliant microbiologist who gave the world the miraculous life-saving drug called antibiotic. But he also had an artistic side that is perhaps less well known. Fleming was one of the first scientists to use microbes to create work of art. He painted ballerinas, houses, soldiers, mother feeding children, stick figures fighting, and many other scenes on petri dishes using microbes. Fleming produced this artwork by culturing microorganisms having different natural pigments on petri dishes to create colorful patterns. Fleming even organized exhibition of his work. And now the innovator has many followers. Painting with microbes in petri dish is called now agar art. Creators use either naturally colorful microbes or genetically modified microbes as paint and various types, shapes and size of agar as a canvas. To create such a sample, the artist must know the words of microorganisms and their physiological and cultural properties to determine the color to paint certain colonies. To draw the attention of the general public to the visible, to the diversity and beauty of the world of microorganisms, various competitions and festivals are held all over the world. The scientists paint landscapes, portraits, and even reproduce famous painting with microbes, as you can see on this slide. On the next slide, you see the winners of agar art contest in 2021. So you can see this beautiful bio art. Bio artists, thanks to modern technologies, create not only paintings or new types of plants, but also music. Researcher Joe Davis created an audio microscope which translated the movement of the harnesses and microvilli of bacteria into an optical signal and then turned it into sound. He discovered that different types of microorganisms sing differently. But his most famous musical bioart project was bacterial radio, for which the artist received the Golden Nick of the Ars Electronica Festival. I want you to listen some sound. Tak, zaraz. Now you are listening the sound of the spike proteins of COVID-19. Translating proteins into sound gives scientists another tool to understand and design proteins. The musician and MIT professor Marcus Buehler develops artificial intelligence models to design new proteins, sometimes by translating them into sound. In the music they created, they analyzed the vibrational structure of the spike protein that infects the host. Understanding these vibrational patterns is critical for drug design and much more. So let's go to conclusion. The analysis of new types of artistic practices, for example, scientific art and bioart, prompt us to pay attention to several points. First, if the author does not have the necessary knowledge, he becomes an ideological inspirer rather than his creator. And artists need not only talent but also competence and a high level of responsibility. Secondly, the widest introduction of the computer, the internet, digital technologies into the life of an ordinary person starting from his early age significantly changed the entire mental and psychological structure of the individual, reorients him from the traditional cultural and civilizational experience to fundamentally different one, far from everything with which man had to deal in a considerable period of history. Hybrid art is not about an artist or scientist, but it emphasizes technological artistic forecasting, technologies and their impact on man and his world, the future that the artist scientist contemplates in his work. This approach to art is clearly illustrated by the works of hybrid art related to biorobots and other forms of combining the living and the inanimate. And the synthesis of art and technology is the empiric way to popularize scientific achievement and ideas, and also is a specific tool for formulating problem and finding ways to solve them in the model world. So thank you for attention. I finished.
Thank you. It was really a very interesting presentation about hybrid art. It was a really thorough presentation. I would like to understand that in hybrid art, so what are different challenges that technology person need to handle? Like if we are finding out the vibration pattern, then what could be the different challenges they need to face?
I think as a result of hybrid art, this type of art recognized principally among artists, often remaining outside the attention of representative of the scientific community. And closer collaboration between artist and scientist is associated with even greater challenges related to rethinking the exceptionally positive role of the latest technologies in modern society, and revising the ethical, political, economic, and other consequences of use of technology for humanity and for the planet.
Okay. It was a nice presentation. That's it from my side. Any questions from the audience?
Thank you so much for the kind presentation, ma'am.
Thanks.
We are our next presenter is David, and the paper title is a conceptual design and early prototypes of a gamified virtual reality interview training application.
Hello, everyone. My name is David Jennings, and I'm a student at Elon University in Elon, North Carolina. And I'll be discussing my research on creating a blueprint for an application in VR that utilizes gamification to help with interview training. So there has been previous research in both the realm of gamification and virtual reality when they are used to help with education, training, and other simulations. For example, there was research conducted with a VR security training, and it produced effects where many of the users said that it helped them with their training. And there have been other instances of research in education where VR has greatly increased user interaction with the topic and their learning. And so what my fellow researchers and I are trying to do is create a VR application that helps with interview training and uses gamification to also, help with it, as gamification has been shown in previous research to help improve user interaction and motivation. There are other existing interview training applications, such as Virtro and Virtual Speech. However, those applications don't focus on gamification. They don't really have many gamification elements. And one of them, Virtro, is not even in VR. However, they have been shown to be helpful towards other users who have used those applications for interview training. Those applications also only focus on the interview itself. With our application, we are trying to cover the broad idea of interview preparation. So our application will help interviewers learn how to prepare for an interview before they ever go to it and even to after it. And I'll go over that more. So our paper was focusing on the conceptual design of this application and our future plans with it. In order to create the application, we had to first identify what elements of interviews we had to focus on. And we were able to come up with 6 core interview aspects, which are, there are eye contact, preparation, time management, question answering, question asking, and follow-up. Each of these core aspects we're going to use and target in our interview training application by using 6 stages. And these stages will form a timeline from preparing the morning of your interview towards completing your interview and then following up.
Sorry to interrupt you. Do you have presentation? Then you can share that also. So it will be easy to understand. Okay, fine. Then you can continue.
Yeah. And so the first stage of this application will take place in a bedroom setting or a home setting where the user will be able to select different items to help prepare for the interview, like clothing items, other necessities like papers, and they'll be tested on what to bring, and what will be useful to bring, and each choice they make will provide a certain number of points. The point system is our way of implementing gamification so that the users will strive to collect as many points as possible. And then after this first stage, the user moves to the second stage where they will be moving throughout an open area from their home to the location of the interview. And in this stage, the user will have to use teleportation, which is a feature provided through VR and how they will move about. And it's one way to introduce them to teleportation, which we use throughout the rest of the application. And it'll also test their time management with trying to reach the interview location on time. And again, the speed in which they reach the interview location will have an impact on the points they earn. The third stage will also affect time management, where it's similar to the second stage, but it's in a more enclosed lobby area of the building where the interview is going to be in. And they will be facing different distractions and obstacles that they have to pass. And again, there's a time limit, and the faster you can do it, the more points you will earn. And then the fourth stage will be focusing more on preparation and learning about the interviewer, the job, the company. And they will be in a waiting room where they search around and interact with different objects, where they get to learn pieces of information about the company. And with each item they choose to interact with and learn about the company with, then they'll gain more points. And it's another way of motivating people to go out and learn about the company before they ever reach the interview. And the fifth and longest stage will be the interview itself, where the user will be asked questions and they will have multiple choice responses on how to respond. And there will be best, good, and then irrelevant options. And then the user will also have a chance to ask questions where they have multiple choice options to select a question to ask. And they will be given points based on how well they choose their answers and their questions to ask. The stage will also challenge their ability to keep eye contact, and they will be recorded in how much they look at the interviewer and be given points based off of how frequently they're able to keep up eye contact and stay engaged and focused. The last stage of the application is about follow-up, where the user will be back in the home setting and they will have the opportunity to send a follow-up email by interacting with a computer. And if they do this, they'll gain more points. And if they don't, it will reflect on their final score. Now, the user will know what to do because before each stage, there will be a list of goals and instructions on how to complete that stage. And they will also always have access to seeing those goals in the top right and how many points they're earning throughout that stage, so that they can keep track of their progress and give instant feedback. And then at the conclusion of each stage, there will be a feedback menu where they receive information based off of how well they did, how many points they received, and what to maybe work on if there were areas where they didn't earn points from. And then between each stage, there is also a comfort check, because using a VR headset for prolonged periods of time can lead to nausea and ocular discomfort, which we are trying to avoid. These comfort checks will ask the user to evaluate whether or not they feel okay, if they need to take a break, and then they'll be able to make a decision there on if they should rest for a bit before continuing. Now, after this application is developed, we're going to hold user studies where participants will use a VR headset to use this application. And then they will also have an EEG headset, so that we'll be able to record raw brain data and learn more about how they're feeling while using the application, like their interest, their stress levels, their excitement, so that we can get objective data regarding how they are reacting to each stage. And alongside this, they will be taking a survey, which is a modified acceptance scale developed by Vanderlyn. And by using and comparing the subjective and objective data that we collect, we can determine which aspects of the application are beneficial towards the user, which aspects help them prepare for it. Do they like the gamified aspects? Do they like all the stages? Are all the stages satisfactory? We'll be able to learn all this information and then better prepare for further applications. And this information could be used for future developments where, for example, if the point system doesn't work well, that can be taken into consideration for a future interview training application or training application in general in VR. And they can just learn from this experiment. And the idea is to create a very broad interview training app where you'll be able to train yourself for interviews, not just the interview itself, but before the interview, preparation, getting there on time, and then after the interview. Because again, as I mentioned, there have been other interview trainers, but they are not as comprehensive as what we're planning to do. And I would like to thank the conference for this opportunity and thank everyone for listening.
Okay, it was a really nice presentation. You have presented your application. My question is that, is there any live interaction between the interviewer, or it is the static hard-coded basis?
We're going to be using a hard-coded interviewer, so there's not a live person you're interacting with. That way, it'd be easier for users to actually use the application, so you don't require someone else to be online to interact with. And this other interviewer will be a simple avatar that's hard-coded.
And my question mean to say that it is hard-coded stuff, or are you applying any machine learning concept over here to, if I'm asking one question based on the answer of the person, I can modify my next question. Like if I'm asking one easy level of first question is easy, then if the user is giving good answer for that question, then my next question might be little bit tougher level. So are you doing that, or do you have some fixed set of questions?
We have a set of questions that we're going to write in hard code into the application. There won't be machine learning to have the AI like change its interactions based on your previous responses, because that could greatly increase the length of the application and lead to issues with nausea and discomfort and other symptoms of cyber sickness if they're in the VR application too long.
Okay. And may I know on which platform you are developing this product?
Oh, we're using Oculus. We are making the application in the Unity game engine with the Oculus integration package. And we're going to be testing with an Oculus Quest 2 and using an Emotive EEG headset.
Okay. Can you just tell me what are different evaluation parameters you have selected to evaluate your complete application? Like it may be possible that there are different application as well. So how you will evaluate that up to which mark our application is better or efficient, we can say.
Could you repeat the question one more time?
Means which type of different evaluation parameters you have selected for evaluating your application. Like it may be it is taking very less time to give a feedback, or like this type of parameters.
Oh yeah. Well, we're going to have the users respond to an acceptance scale where they'll be asked about each stage and whether they thought it was satisfactory, if it was helpful, if they learned anything, and we'll be trying to determine like which stages are more helpful, which stages may have been considered useless. And we'll be trying to use that information to determine in the future what could be used in other applications. For example, if one of the stages, the users respond poorly to it, it has too high stress levels from the EEG data, or if they respond poorly saying that they didn't feel they learned anything from it, then we will, it will give sign to.
Please unmute yourself. Yeah.
Wait, can you hear me? Are you able to hear me?
Yeah, I can hear.
Oh, okay. So we will be using those responses in that data to try to determine which parts of our application are successful and can be used in future applications or future VR developments. And our goal is to get around 20 participants. And so if a majority responds negatively, then that shows the ineffectiveness of certain stages of the application and what we need to work or improve upon.
Good work. All the best to you. That's it from my side. Audience, if there are any questions.
So we are moving our next presenter, Jonathan, and the paper title is the Automatic Control System Based on Industry 4.0 PLC and SCADA.
Good day. I don't know if you can see me from that side.
Yes, sir.
All right. Thank you so much for this opportunity. Let me start by sharing my screen, if you may allow me to do so. Yes, can you see the screen?
Yes, sir.
All right. Okay. So as it shows on the screen, so the title of my paper is Automatic Control System Based on Industry 4.0, PLC and SCADA. And then myself, I am Jonathan William Jackie Bulungu. So let me just quickly go to the next slide. So I am the author of the paper, and I am a mechanical engineer by profession, and I hold a Master's of Philosophy in Mechanical Engineering. And I am from the University of Johannesburg, which is in Johannesburg in South Africa. And myself, I am originally from the Democratic Republic of Congo. And then the co-author is Opeyolu Timothy Laseinde, who is an associate professor at the University of Johannesburg. He was also my supervisor for my master's degree. So my research topic has two most parts, which is Industry 4.0 and automation. And we know that Industry 4.0 is our fourth industrial revolution that comes after the third industrial revolution, is a new trend toward advanced automation and data exchange between industrial processes and technologies. And then we know that Industry 4.0 technologies are cyber physical system, Internet of Things, Internet of Service, big data, analytics, augmented reality, automation, and so on. And then automation is the conversion of controlling industrial processes to a most automatic process and system. And then we know that the automation pyramid represents the automation technology of modern industry. And as we can see, we have the automation pyramid here. We have the we have the 5 fields of the automation pyramid. We have the first field, which is the feed level, which got sensors and actuators. We've got the second one, which is the control level, which got PLCs and other control devices. And the third level of automation pyramid is this it is supervisory level, which got SCADA and HMI, and then followed by the 4th and 5th, which is planning level and management level. For this paper and for this research, I'm mostly focused on the 3 first level of automation. And then we know that from the PLC, from the field level, which we have sensors and actuators, they are input devices and they are information carrier, and they send all the data to the control devices, which are the PLC, which control all the field level. And then from that level, we have the supervisory level, which is the SCADA, which monitors and supervise all the field level. So the objective of my research is to demonstrate the efficiency and the reliability of a PLC SCADA-based system using Industry 4.0 technology. And then for the problem statement, we know that relying on manual processes or semi-automatic processes tend to slow down the operation of an industry, and then tend to cause slowdowns and backlogs in the system. So with using Industry 4.0 technologies combined with PLC and SCADA, we make the system more reliable, more efficient. And then we know that this research is essential to support the development of Industry 4.0 system, and also to provide necessary information for the development of Industry 4.0 automation. So for my research, my research was based on an existing small-scale milling plant, which is present in one of the laboratories in the University of Johannesburg and in the mechanical engineering department. As we can see, this one is a small-scale steam plant, which has 3 subsystems. And then we've got the first one, which is the water treatment plant, as we can see on this picture. And then the second one is the steam generation plant, where the steam is generated And the third one is the steam distribution system, and how it shows how the steam is distributed in the system. The existing plant is semi-automatic, and it also shows a lot of issues and problems which needed attention to be more effective. So for my research, I wanted to develop or to propose a new system that can fully, that can automatically control this mini steam plant. And then for the methodology that are that are used for this research is based on a research framework which is similar to the engineering design processes and also to the steps followed to meet the Industry 4.0 requirements. And then the first step of my research framework is observation. So I observed how the plant was working. I managed to detect what was wrong with the plant, and then from there I went to the next stage, which is collection of data. And then I collect data from first of all observing the plant, and then from the lab technician, the one who is working on that plant, and also from doing some literature review research, and that's how I was able to collect data. And then to develop my proposed design. So it was based on the problem that I saw on the existing steam plant. And then based on those problems, I was able to generate possible solutions. And then from those solutions, I used a decision matrix tool that enabled me to select the best solution and then implement those solutions. And then the fourth step is the testing of the model, which I used a PLC and a software, and a PLC software and a SCADA software, just to simulate and see how my proposed design was going. And then I went to the third one, which is the analysis. How I went to analyze my result and then see how the existing system is compared to the proposed system. And then I went to the conclusion. As you can see on the screen, we have my proposed automation system. We've got the process flow diagram, which shows how it is treated from the first subsystem of the mini steam plant, which is the water treatment plant, and then it goes to the steam distribution plant. As you can see, I've implemented solenoid valves, which were not in the existing plant. I've also implemented sensors, different sensors like the temperature sensors and pressure sensors in the boiler, the float sensor in all the vessels, and also the steam detector vibration sensor for the feed for the boiler fill tank. And then we'll also see the automation architecture, the lower picture. As you can see, we've got your sensors and actuators, which are controlled by a PLC, a S7-1200 Siemens PLC. Then we go to a SCADA. We've got a WinCC SCADA, and also we have a cloud, which enable us to access the data of the plant from a remote location. And that cloud can also send a message to the operator if there's anything that is wrong with the plant. This is the PLC simulation result. I did the simulation using Logosoft Comfort V6.1, which is a Siemens software for PLC. So I use that to simulate my proposed system. So I use a ladder diagram, which is a PLC programming language. So I use that to simulate and see how my proposed design is going to react. And this is the next picture of my PLC simulation. Let me quickly go to my SCADA simulation, which is this one. I use Wonderware in the Soft Web Studio 8.1. So my SCADA simulation was mostly for the condition-based analysis using PLC and SCADA, which are focused more on the boiler and also on the feed pump. As you can see, I was monitoring the pressure, the temperature, and the water level of the boiler. And then if the pressure or temperature or water level exceeds the predefined value on the screen, on the SCADA screen, which will be from in the control room, the operator will be receiving different alarms, different messages showing that there's an issue with the equipment. Same with the feed pump. When the pressure and temperature and vibration goes beyond the predefined value, there will be alarms and different messages received that will be sent on the screen. And also from the SCADA screen, we can analyze the data using the graphs. As you can see, we've got graphs with different colors, which present different parameters. And when the graph changed the color to and went to black, it shows that the parameter has exceeded the predefined limit. And then from the SCADA screen, the operator will be aware, will know that there will be an issue with the system and it will react accordingly. So to conclude, this study just wanted to demonstrate the effectiveness and the reliability of a PLC SCADA-based automatic control system using Industry 4.0 technologies. We know the results obtained from the PLC and SCADA simulation shows that the proposed design is more effective than the existing one and is also more and also meets the requirement of Industry 4.0 automation. And then for future work, I would like to be able to use the SCADA data because with the SCADA simulation, we are able to get historical data. And then with those data, for future work, we want to implement an algorithm that can be able to read the historical data and then also perform more condition-based monitoring based on those data. And that is the end of my presentation. Thank you so much for your time and attention, and thank you also for the opportunity that you've given me. And I'm also open for questions and answers. Thank you.
It was a great presentation. I would like to know about that is what is the response type of your system? Means after
Sorry,
the fault, after how much time we are getting the acknowledgement that there is the problem? Response time.
Yes, with the PLC simulation, it depends on the input comment. That goes from the input for the input comment. That's when we receive an output result. So by playing with the input comment and by playing with the input comment, we will know when and how the output is going to react. So in terms of time, I'm not going to give you an exact time as it takes like this much of time. But what I can say is just when an input, when a sensor senses a different temperature and different pressure, it's going to send a comment to an output, which will be an actuator, which will be a valve. And then from that signal, a valve will react.
Okay. And as you have explained that from multiple solutions, you have selected one of the good solution. So on which parameter you have selected that this is the one of the good solution?
So I used, as I said, I used a decision matrix, which is an evaluation table. So I use, after generating all the solutions for each problem, so I use that table to evaluate this different solution based on the project objectives. So with my objectives, I could be able to select which solution that will be good for my research.
Can you go to title slide, please?
Sorry?
Your first slide, title slide.
Right, that is.
Next one slide. That is where you have said that it is reliable and efficient. Next one. Next to this, where you have said your problem statement. Yes. So here you are saying that to demonstrate reliability and efficiency. Among these two parameters, in which part you are saying that this is reliable and why you are saying that this is efficient?
Okay. So as I mentioned earlier on, the existing system is semi-automatic. So That existing system is operated by someone who has to stand in front of the mini steam plant and press any buttons or open any valves while operating the plant. But with this new system, with the proposed system, everything is automatic. So I used sensors and actuators, which are valves. So what the operator will be doing is just sitting on the control room and then looking from the screen how the plant is operating. So it should be efficient in the way that whenever there will be an issue with the system, an issue with the problem, it will be informed through the SCADA screen that there's an issue. Let's say, for instance, with the boiler, when the pressure goes beyond the predefined value, it will receive an alarm or a message saying that there's an issue with the boiler, and it will automatically open a relief valve to let go all the steam. So that will also prevent the operator to stand in front of the boiler and then open manually the valve to release the steam. So this is one of the efficient process that has been developed.
It was a good presentation, Jonathan. All the best. That's it from my
Thank
side.
you. All right, thank you.
Thank you so much for the kind presentation, sir. All the presentations are done. So I sincerely thank our all authors for their great, excellent presentation in this session, and all our participants for being a part of this World Conference. I hope this session was informative enough to all. On behalf of the whole team, thank you for your support in this version and the previous 5th version of the conference. So we will happy to have you in the 7th version in 2023. So all the presenter would be getting the digital certificate through email within 2 working days. Further, all the papers have already forwarded to Spinger. The publication will be live within 6 months. So kindly cooperate with the team of WorldAce for 2022. I also thank our chair of the session, Professor Gitanjali Shinde. Here is the token of appreciation on behalf of Global Knowledge Research Foundation and the team WorldAce Format. Thank you so much for the valuable presence and the kind information.
Thank you, Riddhi.
Yeah. Can you share some about presentation, ma'am?
Yes. All the presentations are really interesting, and not many in different areas the scholars are working. One of the interesting fact I need get to know about the hybrid art, that was the interesting to know, and the presentation about Jonathan, that was really interesting, as they have already simulated the stuff. So I just like to tell that all the best to each and every researcher. Keep working, and thank you.
Thank you, ma'am. So, dear all, I kindly request to all, please turn on your camera for a quick snapshot of this virtual conference. Thank you so much for the presentation, and thank you so much. Stay safe, take care.
Thank you.
Thank you.